Hey, good afternoon, everyone. Good morning or good night if you're watching this in morning or night, respectively. So yeah, I'm your teacher for today. Basically, I'm gonna teach you biology about vectors for cloning genes in plants and animals. So basically, this is like a part two for the last video. So if you haven't seen the last video, you can check that out. It's in the biology playlist of our channel. CHSC tutorial. Okay, so now we're gonna learn about vectors for cloning genes in plants and animals. You may be surprised to know that we have learned the lesson of transferring genes into plants and animals from bacteria and viruses, and we have known for this for ages how to transform eukaryotic cells and force them to do what the bacteria or viruses want. For example, Agrobacterium tumefacinus, a pathogen of several dicot plants, is able to deliver a piece of DNA known as tDNA to transfer normal plant cells into a tumor. Well, tumor it's not a good thing for layman terms, alright? So tumor is basically abrupt cells which though they do not harm in any kind, but as you say, excess of anything is basically harmful. So yeah, that's how basically the whole concept of the cancer works. So yeah, the tumor cells to produce the chemicals required by the pathogen. Similarly, the retroviruses in the animals have the ability to transform normal cells into cancerous cells. A better understanding of the art of delivering genes by pathogens in the eukaryotic host has generated knowledge from the transfer these tools from the pathogens into useful vectors, delivering genes the interest to humans. I'm gonna break it down for you, all right? So it's basically tumor is like the man-made in the eukaryotic cell. We tested that, and the bacteria's name is Agrobacterium tumefacinus. So we basically what we put a pathogen, and it did deliver a piece of DNA which is known as tDNA, and it basically like transferred the normal cells into like the tumor. Tumor is nothing but same cells over and over again, like the concept of cancer. See, it's written out there, cancerous cells. Well, cancer, if you talk about cancer, it's not like that cancer is like affects like normally in human body. What it does is it grows. I mean, by grows I means it really grows bad. And basically in human body, when cancer occurs, it's not about humans but any other like animals like a dog or a tiger, or even though in plants maybe. So when cancer happens, it basically it does like it regenerates cell. It's over and over and over again, right? See, just think about that. You have a hand, you have the skin over your hand. What if the skin stretches, just never stops growing, just do not stops growing? It's gonna grow. There's no bones, no muscles in that, but still the skin's gonna grow. It's in the end it's gonna be like baggy. That's why it's the reason why cancer is dangerous. It's not like it's venomous or toxic or something. Okay. So the tumor, including TI plasmid of the Agrobacterium tumefacinus, has now been modified into cloning vector, which is no more pathogenic to the plants, but is still able to use the mechanisms to deliver genes of our interest into a variety of plants. Similarly, retroviruses have also been disarmed and now been used to deliver desirable genes to animal cells. Okay, competent host for transformation with a recombinant DNA. See, DNA is hydrophilic molecule. It cannot pass through cell membranes. Why, you ask? In order to force bacteria to take up plasmid, the bacterial cells must be made up of competent DNA. This is done by treating them with a specific concentration for a divalent cation, such as calcium, which increases the efficiency with the DNA enters. Okay, so the bacterium through the pores and cell wall recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice. followed by placing them briefly at 42 degrees Celsius, heat shock actually, and putting them back on ice. This enables the bacteria to take up the recombinant DNA. This is not only way to introduce the alien DNA into the host cells. In a method known as microinjection, recombinant DNA is directly injected into the nucleus of the animal cell. In another method, suitable for plants, cells are bombarded with high-velocity microparticles of gold tungsten coated with DNA in a method of biolistics and gene gun. Well, I'll talk about gene gun later in this video. And last method uses disarmed pathogen vectors which were allowed to infect the cells, transfer of the recombinant DNA into the host. Now that we have learned about the tools for reconstruction recombinant DNA, let us discuss the process of facilitating recombinant DNA technology. Okay. So in this video, you can see the DNA that separates out can be removed by spooling. We'll talk about spooling later, what spooling actually is and what spooling does. The process of recombinant DNA technology, isolation of the gene material. All right. So it's basically we can isolate the gene, the required gene we need, in three processes. Like it's like 3-stage, but I'm gonna break it down for you easily. Okay, so in majority of organisms, this is deoxyribonucleic acid or DNA, and in order to cut DNA with restriction enzymes, it need to be in pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with other macromolecules such as RNA, and RNA is ribonucleic acid. Okay, ribonucleic acid, and DNA is deoxyribonucleic acid. Just remember that. For like if you make this so far, you probably know what DNA and RNA is from like literally from 7th class and 7th grade. So but it's India, hey, we don't know what to expect. Okay, so lysosome, bacteria, cellulase, plant cells, and chitinase is basically a fungus. Okay, you know the genes are located on long molecules of DNA intertwined with proteins such as histones. The RNA can be removed by treatment with ribonuclease, whereas proteins can be removed by treatment with protease. Other molecules can be removed by appropriate treatments, and purified DNA ultimately precipitates out after the addition of chilled ethanol. This can be seen as a collection of fine threads in suspension. Okay, so cutting of the DNA in specific locations. So it's basically in what pinpointed location we need, of what particular gene we need in the DNA. We're gonna separate it from the for the preparation of the recombinant DNA. So amplification of the gene of interest using PCR. See why we need amplification? Because process is gonna fail. We have no surety over on such a micro level. So we're gonna make sure that it's basically we make enough copies so that we would have strong data to back out our results. Okay, PCR stands for polymerase chain reaction. In this reaction, multiple copies of gene or DNA of interest is synthesized in vitro using two sets of primers, small chemically synthesized oligonucleotides that are complementary to the regions of DNA. An enzyme, DNA polymerase. Sorry for that. So DNA polymerase, yeah. If the process of replication of DNA is repeated many times, a segment of the DNA can be amplified. Okay, so, geez, just stop texting me. Insertion of recombinant DNA into the host cell organism. There are several methods of introducing ligated DNA into recipient cells. After making them competent to receive take up DNA present in surrounding. So if a recombinant DNA bearing gene for resistance to an antibiotic, e.g. ampicillin, is transferred into E. coli cells, the host cells will become transformed into ampicillin resistant cell. If you spread the transformed cells on agar plates containing ampicillin, only transformants will grow and untransformed recipient cells will die. Since due to presence of ampicillin, the ampicillin resistance gene is called selectable marker. Obtaining the foreign gene product. When you insert the piece of alien DNA into cloning vector, the transfer will into a bacteria. Okay, so it's called well, basically, if any protein encoding gene expressed in heterologous host is called a recombinant protein. The cells harboring clone genes of interest may be grown on a small scale in the laboratory. The cultures may be used for extracting the desired protein and then purifying it by using different separation techniques. Okay. So this is basically an image of bioreactor. This is the acid base for the pH control, percent of hydronium ions, steam for sterilization is some water up in there, and foam breaker, and below foam breaker we have flat-bladed impeller, and culture broth, then in the end it comes to sterile air is a place for it. Then in the second figure we see it's basically bubbles dramatically increase in the oxygen transfer area, and that's the gas entertainment, and an increased surface area for oxygen transfer. In B is basically sparged tank bioreactor through which sterile air bubbles are sparged. A stir tank reactor is usually cylindrical or with a curved base to facilitate the mixing of reactor contents. The stirrer facilitates even the mixing of oxygen availability throughout the bioreactor. Alternatively, air can be bubbled throughout the reactor. If you look at the figure closely, you will see that bioreactor has an alligator system, an oxygen delivery system, and a foam control system, a temperature control system, a pH control system, and sampling pods, so that sample volumes of culture can be withdrawn periodically. Okay, and the downstreaming processing. Okay, so it's basically the downstreaming process. After completion of biosynthetic stage, the product has subjected through a series of processes before it already marketing as a finished product. The process includes separation and purification, which are collectively referred to as downstreaming process. The product has to be formulated with suitable preservatives. Such formulation has to undergo through critical trials, as in case of drugs. Strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product. Okay, so I'm going to give you a small summary of the chapter to get a whole overview. All right, let's start. Biotechnology deals with large-scale production and marketing of products and processing using live organisms, cells, or enzymes. Modern biotechnology using genetically modified organisms was made possible only when man learned to after chemistry of DNA and construct recombinant DNA. This key process is known as recombinant DNA technology or genetic engineering. This process involves the use of restriction endonucleases, DNA ligase, appropriate plasmid or viral vectors to isolate and ferry the foreign DNA into the host organisms. Expression of the foreign gene, purification of the gene product, i.e. the functional protein, finally making a suitable formulation for marketing. Large-scale production involves the use of bioreactors. So yeah, basically this is the end of the first chapter of the biotechnology unit. I'm gonna talk about the NCERT and NCERT exemplar exercises in the next video if you want to. So make sure you write down the comments your doubt below, and subscribe and like to the YouTube channel CHC Tutorial. Okay, so apart from that, if you got any doubt, you can contact me personally at the rate ig.weed, it's ig.we3d on Instagram. Okay, so do not forget to like, share and subscribe, and do not forget to comment your doubts below. if now let's keep up for a aim. If this video goes 100 likes, I'm gonna do a live session for exercises for NEET or UAT and NCRT exercises upon your choice. All right, so make sure you do not forget to like, share and subscribe. Thank you, and see you in the next video. All right.